So hi everyone and welcome to this week's seminar, and it's my great pleasure to introduce our speaker today. It's Laura Lüthy, and she's visiting the EBD and especially to work in our group, hence me introducing her. But she's visiting us from the Max Planck Institute for Animal Behavior in Constance, where she's a PhD student currently, and how their behavior and population dynamics are changing under climate change. And she'll be talking about this for the next 30 plus minutes. And I know Laura quite well because she interacted our research in Zurich, in the University of Zurich, where she did her master's and where she worked on the behavior of meerkats. And so she has a lot of experience on working on animal systems and their behavior in responses to climate change. And so for all of those interested in interacting with her and talking to her and getting to know more about this wonderful study system, come visit her in the EBD-2 in the Green Building. She's there on the first floor in the fishbowl big office. So anyway, without further ado, I give the floor to Laura. Thank you.
Thank you for the introduction, Maria. So I'm going to talk to you today about the colobus monkeys and climate change. And climate change affects all our environments, not just the polar and the desert regions, but also the tropics where most animals of the planet live. And those animals also are not as used to big variations in temperature, and so they will really experience unprecedented conditions, like many on our planet. And this is also the case for the endangered colobus monkeys, which you can see here. They live in the tropics in Uganda, and they're already endangered, and unfortunately the most individuals in this population live in one kind of viable population, which is in Kibale. So we know very little about how climate change will affect them, and that's why I study exactly this for my PhD in Constance. So how will climate change affect their environment? Here I have a map for how climate will change in the primate ranges, and the black square is where my population lives. So on the left you can see it's going to get warmer, no surprise there, but it's going to get much wetter as well in there. As I mentioned, they live in Kibale National Park. It's a moist evergreen forest. It's quite high up. It's at over 1,000 meters above sea level, and it's quite hilly as well. With the humidity, it gets quite exhausting working there, honestly, as a European, I have to say. And it's where the largest and most viable population of colobus live. And we are very lucky to have a long-term data collection led by Colin Chapman for the last 20 plus years. The colobus live in multi-male, multi-female groups. They're usually they're quite content with each other. They're social, but they're not in each other's hairs like a lot of other primates are. They're considered an indicator species for a larger faunal decline. So they're usually the first one, especially of the primates, when the habitat is disturbed. Also because of hunting, they're quite large. They're up to 10 kilograms for the males, sometimes a bit more. And they don't really run away when they're being hunted, which is a bit dumb of them, but that's also why they're going extinct in parts of the world. So they're kind of considered a bit like a canary in a coal mine as well. They can live up to 20 years. I think most of them in the wild don't make it that far. I think females go up to 13 years. We don't exactly know much about kind of all of this because they can't be kept in zoos, probably because of their diet. So everything is kind of approximate. So we know their gestational period is about 5 to 6 months for their pregnancy, and then with their young, the first year they're quite close by, and then they get more and more independent, and they reach sexual maturity around 4 to 5 years old, and then they have their second infant about 1.5 to 2 years later. What's very interesting about them as well is the dispersing sex is actually the females, which is not the case for most mammals. When they reach kind of sexual maturities, the females leave the group. Some of them also stay, which is great for our data collection, so we know more about them. And they're quite tolerant as well. So we do have a few females that came into the group with a very young infant attached to them, so a few days old maybe, which again, if you think about lions or other mammals with infanticide, the males are surprisingly okay with that, at least in our group that we study. They are folivores, so they mainly eat young leaves. They really specialize on that. They also have a specialized gut system, so they have foregut fermentation. Kind of, if you think about cows, to eat the leaves, to be able to digest them, they're really specialized on that. But they also sometimes consume bark, more and more in the last few years, and we don't really know why yet. There's a few hypotheses. It's very interesting, but also because of that folivorous lifestyle, they have to spend a lot of time resting. So a typical day with the monkeys usually is you go out there, they're feeding, and then they're resting for a long time. They defecate, and then the cycle keeps going. So it's kind of like you have to, after resting, you have to kind of be careful where you stand in the forest so you don't get wet. Which I have managed to not get that, but I think I'm one of very few people that work with them. So always wear a hat. And then they keep going, they feed and they rest. Then there's also, it's very known for primates. There's around 12 primate species living in there. There's also elephants, more and more, that live in there. Especially also baboons. Baboons really like breaking into stuff. This is our kitchen. This is my friend watching him. Because of the baboons, we have bars on the windows, not like real glass windows. But they've started just lifting their babies up and have them go into our houses, which is kind of scary when you get into the house and there's a baby baboon screaming at you. But they're getting really sneaky. What they're also known for is we have a lot of chimpanzees in the forest there, which they're very fascinating. But since I've worked with colobus, I like them less and less because they really like our colobus. So they're hunting them a lot. So if you don't know, chimpanzees also eat meat. They're very strategical hunters. And they go for the colobus infants and juveniles and also the females. And in other parts of the forest, they also decimate the population of colobus quite a lot, I think up to 12% per year. So they really go at them. But coming back to the colobus, what I study is the climate change part. So what I first am interested in is the cascading effect, so the direct and the indirect effects. First of all, climate affects food availability. So temperature, a lot of climate variables can affect forest health and productivity. So I wanted to look at if young leaf availability, which is what the monkeys primarily eat, it's like 80 to 90% of their diet is young leaves, if that effect is changed due to climate. But then I also want to know how climate and food availability both affect the behavior. So I here look at activity budgets, how they spend their time. So if you think about it, if it's very hot, you also prefer to go to the beach and lie around and not work as much, for example. Or if it's raining, they like to hunker down and kind of huddle together. So that affects the behavior. But also food availability. If there's a lot of food around, you might not have to travel so far to get the nutrition that you need, or the food is better. And then all of this together can also affect the population dynamics. So climate, but also climate through food availability. If it's a very good year where there's a lot of food around, they might have more offspring, but also through the behavior, how they socialize with each other. Sorry. So this is what I'm looking at for my PhD. The population dynamics I'm looking at with Maria here. So today we're going to talk about the first two parts here. So we're very lucky to have a long-term data set led by Colin Chapman. So we have 23 years of phenology data, of tree phenology, and 15 years of behavioral observations of these monkeys. And so we know what they're doing, who they're doing it with, and what they are eating as well. And now the first questions for climate effect on food availability. I wanted to know what the seasonal and long-term patterns are, because we don't actually know in that forest. And then I also wanted to know which climate factors best predict these patterns. And then also if EVI, which is similar to NDVI, it's a remote measure of greenness, can be a good proxy for what we're doing in the field every month, like on clipboards. And for that, we use monthly tree phenology. So every month, a field assistant goes out and measures how many young leaves there are. So they look at the tree crown and determine how much of the tree crown is young leaves. So it's a scale from 0 to 6. And the same they do with fruit and mature leaves. And then we also have transects where they measure the forest, what species are in the forest. So they have transect where every few years they go around and measure what species there are. Because they're also very specialized on the species they eat. They really prefer, at least our group, one species, and then they substitute it as well with other things. For the climate variables, we have local precipitation. We also use temperature data, cloud cover, atmospheric CO2, surface solar radiation, and ENSO and IOD. And ENSO is the El Niño index, you probably heard about that, and IOD is the Indianic Ocean dipole, which is similar to the ENSO, but more impactful for the Eastern Africa. And then the EVI is the Enhanced Vegetation Index, as I mentioned. So it's very similar to NDVI. It's a measure of greenness, but it's more sensitive, which is very good, and it's also been shown to work in the tropics. So with NDVI, unfortunately, it kind of plateaus in the tropics because it is very green. But we know that for EVI, it kind of is more sensitive and it doesn't plateau as fast. And just for fun, I'm going to show you what EVI looks like in Africa throughout the years. So we can see an actual shift even in the central tropics and also in Kibale, which is that little spot. So we do know it does shift at least a little bit. For more technical modeling, I used generalized additive mixed models with two spline terms. So one spline is a seasonal spline, cyclic seasonal spline for the seasonality, and one is kind of a long-term trend spline. And then, so we know kind of the patterns, what they fit the best kind of model. And then we get a seasonal spline out of that. This is, for example, rainfall. We know the seasonality. We know the trend, what's happening. And then together with the intercept, that gives us the model predictions. And for this project, for each climate variable. And then we also did that for young leaves of the 12 most preferred species, which then we replaced one of the splines with a climate variable to see which model best approaches that kind of perfect seasonality. So for example, we would replace a seasonal spline with rainfall and compare the models with the ELPD, which is kind of like an AIC, to see which models basically come closest to the perfect seasonal and trend spline models. And then with that, per species I had 82 models, which I'm not going to show you all of them. So for rainfall, we have a clear seasonality. There's two rain seasons in Uganda. We have a smaller and a bigger one. They peak around April and October. And then for temperature, there's a peak at the beginning of the year, and then it drops down with a minimum in July, which is also the driest month, and month with the least solar radiation, and the coldest month. And these are the best predictors for seasonality. I'm going to take that away, so I'm not going to show you all the climate variables today. For long term, the most important predictors were atmospheric CO2, which we all know is rising, and then solar radiation, which kind of is related to the solar cycle. And we see over the long term, it kind of drops down until 2005. There's an increase until 2018, 2019, and then it starts dropping again. And for seasonal patterns for the young leaf community, we also see two peaks for the seasonality. So you have the month of the year on the x-axis, and like higher is more young leaves, basically. So we see the peaks are also in April and October, and if you paid attention, it's basically the same as the rain season. So during the rain season, the young leaves actually peak as well. And we do have some variation with the different species, but all of them have some kind of seasonality, or almost all of them. Then for the long term, so this is the prediction plot. The squiggly line is actually the two peaks that you see within the year. And it's very nice. It kind of drops down until 2005, 19, and then it starts to decrease as well. There's an overall increase that is very good correlated with CO2 in a lot of species. But what is really nice is it really matches the pattern of solar radiation as well. And we've submitted this manuscript, and it's also available as a preprint. But usually we only know that solar radiation is really important for seasonal young leaf production, but we haven't really looked at the long-term effects, especially in the tropics. And then, what about EVI? On the top, again, is the seasonal pattern that we collect on the ground, and on the bottom, it's the seasonality of the EVI. And the EVI also reflects that kind of biannual pattern as well. So we have a peak in April and a peak in October, and it's also a significant correlation between them. There was less correlation in the long-term patterns. Kind of the trend going upward is shown as well. But what we see in the analysis as well is that it doesn't reflect the variability as well. So for seasonality, it was great. For the long term, it wasn't as good. So to recap quickly for the first part, we had 2 peaks during the rain seasons. The long-term leaf production was really well reflected by CO2 and solar radiation. And EVI was a good predictor for seasonality, but less so for long-term part. So now that we looked at the young leaf availability, I'm going to move on to behavior and special activity budgets. And I thought I'd show you, even though colobus monkeys are sometimes a bit said to be boring and not as social as other primates, they do sometimes play a lot. And that's why I was going to show you a video, because they're not as boring as everyone says they are. And they also tug at each other's tails a lot. They're also said to be able to jump up to 50 meters. They can jump quite wide between the trees. I was surprised to learn that they can fall from 20 meters and be more or less okay. We identify them a lot with their tails because they have kinks in them. So we have a left bent tail and a right bent tail and things like that, which is useful. Sometimes I wonder how they're doing. So for activity budgets, I mentioned them, but what are activity budgets? So it's very simply put how much time you spend on what behavior. And time is a limited resource, as I think we all know. And it's very also linked to energy management. So we kind of know also with climate change, it's a really good window into behavioral strategies and flexibility. So if it gets hotter, what are their reactions? Maybe also what are their limits? And where do they have to kind of make cutoffs on other behaviors, like sociality? So if we think about it, a limited resource, a really nice pie chart. So they spend a lot of time feeding and resting. And this can be influenced by climate, if it's hot, if it's raining. So I mainly looked at temperature and rainfall effects, how they directly affect the activity budgets. Then also diet, what you eat, what you can eat. And this is then obviously for here the food availability, and that's again affected by climate. But what's also important is reproductive state. So males and females, for example, have different nutritional needs. The males are quite a bit bigger, but the females get pregnant, they have to lactate as well. It requires a lot of energy. And also it will probably shift their diets. When I was thinking about this chapter, I was thinking of kind of pregnant women with cravings. So I thought maybe the females, the pregnant ones, also shift their diets. So we'll see. So for the research question, first I wanted to look at just general differences between sexes and reproductive states, and then how temperature and rainfall affects them, how food availability changes them, and then how the diet differs. And for that, I used 15 years of behavioral observations. I used scan data, which is when a researcher goes out, it's kind of a type of behavioral observations, and they kind of every 15 minutes or 30 minutes, they pick out 5 individuals and write down what they're doing and who they are. So they write down individual 1 is feeding, and then individual 2 is moving, something like that. And then when they're feeding, they note down what they're eating, so the species and the part of the plant. And then when they're interacting with someone, they say with who, and the nearest neighbors, etc. And we have 18,000 scans, which now have 93,000 observations, which is quite nice. And I kind of binned that into the behaviors feeding, moving, resting, and socializing, because they're what makes up most of their activity. And as I mentioned, we can identify the individuals, which is great, and they also stay in the group for quite a while. So for the females, we have around 200 births that we recorded, and because we go to the group so often, we quite know well when the individual was born, and from that we kind of calculate. So I took 6 months because we know it's between 5 and 6 months probably, and I binned that into early pregnancy, which is 6 to 3 months, and late pregnancy 3 to birth. And then for lactation, I took the 1 year for being lactating, because we know they're quite often lactating then. They can still do so later, but just occasionally. And then often another individual is born as well after a year or 1.5 years. And then I used Bayesian multinomial models to analyze this. So the plot is, on the left is always the predicted activity, so in percent, and then on the bottom first is the males, then the non-reproducing females, so they're neither pregnant nor lactating, then the early and late pregnancy and the lactating females. And here we can see the males, they feed and rest around 43%. That doesn't leave much room for something else. It's more than 85% of their activity. For the females, they are a bit more differential. They rest much less than the males, and they feed more. And it's kind of also a pattern of just the more differentiated behavior. So they generally feed more than they rest. And that's also the case for the late pregnancy and the lactating females. And then we also have movement and socializing. And social, for the colobus, is basically any interaction with another individual. So it can be agonistic, but also antagonistic. So chasing is also in there. And then moving is about 9% of the behavior for the males, and very little socializing or chasing. And for the females, it's a bit more of that. So they have more interaction with other animals, especially than the males. And the pregnant females even more. And the lactating females come a bit down again. For the temperature, here you can see the Tmax on the x-axis, and again the activity on the y for feeding and resting. And we can see the harder it gets, the less time they spend feeding. And then for the resting, the males rest a bit more when it's warmer, but the females don't change as much. And when we add in the early pregnancy females, we can see they have kind of often the same trend as the females, but just a bit more extreme. So the females seem to be a bit more sensitive to changes in environment. Also for the late pregnancy, and then for the lactation as well. And when we move on to moving and socializing temperature, we can see again they're moving a little bit more, but the females really spend more time with other individuals when it's hotter. And again, the early pregnancy females are even more with that. And then as well, the late pregnancy and the lactations. So they seem to just react more to environmental differences. Coming to rainfall, this is a nice picture I took with my binoculars. It's not great, but that's basically, it looks a bit sad, little male. So when it's raining, they spend more time feeding and less time resting. And especially, again, the early pregnancy females, the late pregnancy females, and also the lactating ones, which during the rain season there's always also more food available, but we'll get to that later as well. Moving and socializing, we can see there's less movement, probably also because there's more food around, they move less, and then socializing kind of stays the same. If we add in, again, the early pregnancy females, the late pregnancy do something else. I still don't know why. And then the late is also kind of doing less. Okay, now coming to the young leaf availability. It's a bit a different story. The females spend less time feeding when there's a lot of food, probably because they get what they need faster, but they spend much more time resting. So the females are always feeding more than the males, but with the resting, they really kind of catch up when there's a lot of food. And then adding in the early and the late pregnancies, we can see still a bit of a different pattern, but they do get affected, especially then also the late pregnancy and the lactations. Moving and socializing, there's much less movement when there's a lot of food, probably because they don't have to find new feeding patches. And then the socializing, the males spend more time socializing as well, or chasing other males. would be interesting to kind of look more into that is like agonistic and antagonistic behaviors. And then again, the pregnant females kind of usually have a stronger curve, a stronger slopes. So to recap, it's hot, they feed, they spend less time feeding. The females rest less, but the males rest more, but they're all moving and socializing a little bit more. For rainfall, there's also more feeding and less resting. So they do have enforced resting time, which is their digestion time, but it seems that it's not necessarily the food availability, so what they're eating. So if they eat more, they don't necessarily have to rest more. Maybe it's also because they eat nutritionally more, like higher qualitative food. We don't know yet. And then for the food, the males spend more time feeding and the females less, but they do rest more when there's a lot of food around and they don't have to move as far. So it's kind of a nice time for them. And the reproductive stage really shows a lot of differences. So especially pregnant females, early pregnancy, but also lactating females react much more to environmental changes than the males do, and also the females. And it's quite important since the females are also usually the factor that determines population growth. So now also coming to the diet. This is just a bit of an overview, so you can see the young leaves in red are really their favorite food. And then we have in blue the bark and mature leaves and leaf buds that they eat. If we zoom in for the young leaves, we can see male and females, they both eat a lot of young leaves. But especially the lactating females at the end, they seem to really appreciate their young leaves. So they really focus on that. And when we look at the other diets, so bark at the top, they do like to eat kind of a 10% bark, but the pregnant and especially lactating females kind of don't eat as much bark than the others do. I assume it's probably because of nutritional needs. It would be nice to kind of work on that, but pregnant females have other needs than non-pregnant females or males. Especially lactating, I assume they need a lot of energy, and maybe they prefer the more high-qualitative young leaves because they have different nutrients. And for the bark that they eat, it's mostly the cambium that has a lot of sugar, I think. So I mentioned young leaf availability, how it affects the behavior, how we come back to population dynamics. So I haven't worked on that completely yet, but I can give you a bit of a sneak peek. So Chapman et al. Have made a study for the long term for a lot of primates, and they show the group size of the monkeys. So this is our monkey group as well. And the group size has increased until 2018, but it's started to decrease. It's kind of stabilized. But what's really cool is that when we also look at the kind of the young leaf availability over the long term, it seems to kind of match that. So we'll have a look at that, see how young leaf availability also drives the population dynamics, see if pregnant lactating females have a different impact. Okay, so to conclude, solar radiation, atmospheric CO2 really drove the long-term tree phenology, young leaf phenology. We see very much differences in activity budgets and what they do, how they react, and we do so especially with the females in reproductive states. And so as the next step, we do the population dynamics. So I would like to say thank you especially to our field assistants who are in the field every day in the forest with scary elephants. And they've been the same field assistants basically for the whole study. So we had one person doing the whole phenology. He's been training another person for a couple of years, but it makes it quite nice for us so we have consistency. But I also thank my supervisor, Urs Kalbitzer, and Colin Chapman as well, as well as my co-authors, Patrick, Laura, and Patrick Omecha, and my tech committee, Mark von Kloinen and Arpad, and Maria for inviting me here. Thank you for listening, and if you have any questions, now's the time, or later, in the fishbowl.
Thank you so much for the presentation. Do we have questions?
Thank you, Laura. It was super cool. I was wondering if you also have data on the on the feeding, on the three species in which they are feeding, you know the name of the species that they are eating. When you take the data, the observational data, they also annotate the...
Yes, so we note down exactly what they're eating. So they really like Prunus africana at our place, for example, or Celtis, different species. And the analysis I did for the young leaf phenology is just on those species that they eat.
Only
like those 12, which is like 60% or 50% of the diet.
Yeah, but do you also, maybe you can also see if the response of climate for the young leaves is the same for the 12 species?
No, it's not exactly, but they do have similar patterns. So we have kind of, so if we go more in depth about the species, we kind of have 3 or 4 different patterns that we have, but they're all very similar with kind of seasonality, most of them during the rain season. But there's, if that works. This is, for example, the long-term pattern. So they do have variation for the different species there, but they kind of match quite well.
Okay. Yeah, because I was wondering maybe the response was different for a species, but doesn't look.
I mean, some of them are. Yeah.
Okay, thank you.
More questions?
Hello, great talk. I really enjoyed this. I was wondering, you mentioned in the beginning that like females spend more time socializing. That's one of the things that you saw. Considering that they are also the sex that is moving away and dispersing, is there some sort of social hierarchy that they're trying to establish and that's why there's more social interactions, or is it more childcare, social childcare or something like that? Do you know why they're spending so much more time, or is it just gossip?
So they've done, so a supervisor worked on a social network analysis for them. It didn't really show much. So they don't have their favorite best friends or anything that they work with. There is a social hierarchy. The males are also usually dominant. They're also bigger. But also they mate with them. That's something that happens. Or just general commotion, or it's about infants. So they don't have communal childcare really. They all care about their own kind of offspring. So they're not as much, I would say.
Thank you.
Thanks for your seminar. So you said that in your data set you can identify individuals. So does it mean that you can identify groups of genetically related individuals?
So there's not been genetics done recently. There have sometimes. So we know, for example, some of the males are related with the females as well, or offspring. Most of it is, we know the offspring, so until 2 years old we know which mother they belong to. And then some of them we can identify because they have like a stiff finger or something, or like a pink face. That's how we kind of follow them, but with the relatedness, but we don't know exactly.
So you are not considering kinship in your models, or these kinship relationships. But don't you think that this kind of structure must play a role in all this system?
I think it's important. So what we know, like when they're chasing each other, for example, if there's a subadult male, a female will protect them, the mother, presumably. So we know the kinship of a lot of them, but only the ones that in the group. If they come into the group, we don't know that.
Yeah, thanks for the very interesting talk. I find it really fascinating that you found these links with the solar cycle of this young leaf phenology. But I was wondering, why do you think that this EVI has a different long-term trend? Because this is also plants, and so is this linked to like rainfall patterns, or like what is your explanation that you find this very differentiating associations? Yeah.
So we didn't test the climate variables on EVI specifically. We just see that there's a difference between the long-term patterns between the ground phenology and the EVI. And I think most of what we saw is that the ground showed more variation, like smaller variations, which should be shown in EVI as well. What the explanation I have at the moment is probably as well we should look at, so the community model here is just of the preferred species, and it might be different for kind of the whole forest, the whole area. So we have the tree phenology of Makerere field station around there where our group lives, and EVI is whole Kibale, so there also might be a difference because of that, I think.
Okay, yeah,
But
interesting.
I don't know yet.
Yeah.
Hey, Laura, thanks for the talk. I mean, so I actually had the same question, and I was wondering, is it related to what exactly they're eating, and that maybe as you're spotting or as you're noting stuff in the field, you know, you're not capturing the young leaves of the whole forest, or there's kind of a bit of an like a bias towards the trees that the monkeys are sitting in, or something like that?
So the leaf phenology is definitely biased towards what they're eating. So I think we know quite well what they're eating. The field assistants are really good at identifying when they're eating it. But definitely the phenology is not, so I did the same model with 37 species that we have. And there was still the bimodal pattern and kind of the long-term trend. But what I put in at the end is the leaf phenology for those 12 species that they really eat, if that makes sense.
Yeah, yeah. I mean, I think bias is the wrong word, because I mean, like I used it wrong, because I mean that's what's more important for that species, right?
Yeah. So I just focused on these species, for example. There's also the black and white colobus, which are folivores in the forest. They like a lot of the same species as well, so they seem to be also quite important for the folivore population there. That's why I decided on that. But for the whole Kibale forest, everything that's living in there, insects and all of that, that also has different like weightiness, I guess. What do you choose?
Thanks, that was a really interesting talk. So my question is a little bit related to the kinship question, but do you think there are persistent differences in activity levels of different individuals? And do you think you have enough data, not necessarily you and this PhD, but do you think the data set has enough to be able to tease some of that apart and be able to ask questions like, do individuals with different traits in terms of their activity or their feeding or their social hierarchy have different?
I think maybe. It's a bit of a hard question, I think, to answer like that. So we have individuals, for example, we have a female that's been there since 2006 for 10 years. So we have a lot of observations from there. We also have focal data that as well. So basically, they just follow the same individual for a long time and write down what they're doing for like 10 minutes. So we also have that data. So I think for some of the individuals, we certainly do. But then kinship, some of it is assumed, and some of them we lose them when they're juveniles, when they become subadults, so we don't know who exactly they become.
Right.
So
but if we don't,
if we did genetic analysis, I think we could.
Or what if we don't need to know the kinship, but we just need to know if they're a high activity, like an individual that spends a lot of time moving, or an individual that spends a small amount of time moving?
I think it might be possible as well.
That's cool.
Yeah.
Thanks for the talk. Maybe you explained this before, but I didn't understand this. Your data is at the end is timing that the monkeys are using for different activities. So the sum of such activities is one.
Yes.
So that's it. So they are dependent.
So basically, for the analysis so far, because I'm not quite done yet. That's why it's not published as well. So what I used is I have scan data, and then I know per month individual BBH ate 20 times young leaves and 2 times bark, and then I have kind of the proportion at the end. So I have the total of what's eaten, and I know how much of that is young leaves, how much is bark. Just occurrences, basically.
Okay. Is it because I was thinking that as they are dependent, and when one of the activities are increasing, the other one is decreasing, how these things are affecting the assumption of the models? The
binomial models.
Yes.
But with that, this is correct. Okay,
yes.
Perfect.
It should be.
Well, this is actually not a question for her, it's a comment. So I am the person who did the kinships of these animals. I didn't want to make any questions to her, but just so you know, because this has come up twice, I didn't want to comment on the first time. The behavioral data and the kinship data are almost identical. 99% of the parentage is accurate. So because this has been done for so long, and this work has been done, like the field assistants have been working on the same animals for so long. When we did the relatedness for disease, that is what we were doing, the relatedness, the behavioral data that they had, and the kinship that I got from the microsatellites was 99% accurate. Just so that is useful for the questions that you made before. And thank you for the talk. It was really great to see again the animals.
That's really good to know.
So that's good news, because it means that your individuals are not statistically independent. So there are some sort of relationships that, in my opinion, needs, they need to be considered one way or another, right? So perhaps, using data, you can start, to trace relationships between individuals. So when you put such relationships into these models, right? So like, for example, you can even put, the presence of chimpanzees hanging around with bad intentions, let's see, perhaps you have done that as well, But I'm worried about that, that you are looking at, in my opinion, that's just a mere opinion, right? But you are looking at this system as each individual is totally independent from each other. But behavioral data can be used, to draw some sort of relationship that can affect the model outcome.
It's very true.
That's my point of view.
Oh, that's good.
Thank you very much.
Hi, thank you for the talk. It was very nice. I have a few questions just to put together everything. So what you're presenting is based on the observations. And how much time per day do you observe them?
So we observe them from, I think, 8 or 9 a.m. and then they have a break at lunch, but the monkeys basically take a break as well. Okay. Which is nice for us, mostly they do. And then until 4 to 5 p.m. and I leave them.
Always the same hours, because it's in the area where this is the hours of the light time?
So not exactly, well, it's quite at the crater, so it's more or less 6 to 6, more or less. So they do leave a bit earlier because it's dangerous in the forest in the dark with the elephants. But we always have the same hours, basically.
Okay.
And in the data set I also just took those hours as well.
Okay, and food data is also based only on observations, not on feces analysis or anything like that?
No, it's just observations.
Okay, and when you say movement, it is including movement, it's actually referring to the other question, for example, running away from the predator, or...
So for the predator, it would be a different thing. Also, we don't observe that often, in our group at least. We're very lucky. The chimps are not as bad in our area, for the monkeys at least. So we don't have those incidents very often. And then they also, if they hear the chimps, because you can hear them from far away often, they also kind of sometimes avoid them. And the movement, so it's not running away from predators necessarily, then they make a note of it.
Okay.
And it's basically walking or the jumping, like to a new tree branch, like just general movement. So a lot of the time they just sit and eat, or just sit and rest, or groom each other.
That's convenient.
It's very convenient. Well, it's less convenient because they're in the trees and in the rainforest it's dense, so these pictures are not really reflecting that very well. Okay. But often you kind of just see them more high up as well, but it's quite convenient for us.
Okay. And last question, something closer home for me, because you know that, for example, parasites influence the behavior a lot. Do you have any data about the parasites that they have? Because it also may influence the diet. because they...
No. We do.
Okay.
We also have notes of sometimes they have like also in their face like fungi and things like that. So we have notes about that.
Thinking about changing diet because of the gastrointestinal, for example.
Yeah.
We know the parasites like toxoplasma also changes the behavior, like the bravery, so to speak.
What is also very cool is we know they also sometimes eat charcoal, which can help with different things. And red soils, we don't really know why that is. So we do have records of that as well. But yeah, it can influence the behavior, of course.
Okay, cool. Well,
that's why I'm glad I have a lot of data. So it kind of, I think, evens out.
Good, something, a lot for you to do. Thank you very much.
No. Little time.
Do we have any questions online? Okay. Any more questions from the audience? If not, thank you so much for a great talk, and thanks for the vivid discussion.
Thank you.